So let's talk a little bit about the physiology of neurons and the nervous system. And we're going to start with a quick review of the cell membrane potential issues. When we're talking about cell membrane potentials, what to remember is that all the function of the neuron is based off of the chemoelectrical potential that's established by where membranes are about the plasma membrane. And this is going to lead to polarization. And any change of polarization is going to lead to the functional change or the action potential being sent and possible structural changes to the cell itself based off of rearrangement of the neurofilaments and neural tubules based off of utilization. So how are we going to go about establishing this potential? It's based off of ions about the membrane and the concentration of those ions in between the extracellular compartment and the intracellular compartment. What we have to remember is that at rest, we have a large amount of sodium and chloride in the extracellular compartment and a large amount of potassium, organic phosphates, and amino acids on the intracellular compartment. The difference between the ionic charges across the membrane gives us an approximate resting potential of about negative 70 millivolts. And so if we look at this distribution, remember we have high amounts of sodium and chloride extracellularly, high amounts of potassium, phosphates, and amino acids intracellularly, and the difference between the two is going to be establishing that charge differential, resulting in the resting membrane potential. This resting membrane potential is going to allow us to establish a relative electrical charge based off of the presence and absence of concentrations of cations, positive ions, and anions, negative ions. Where based off of our reference electrode being intracellular, we get a difference across the membrane of approximately negative 70 millivolts. This negative 70 millivolts will change based off of receptor activation, in which we have two distinct types of receptors, and then changes that can take place based off of activation of those receptors. We have what are referred to as metabotropic receptors. These are receptors that are going to be involved with a response to a change of metabolism within the cell. And then we have ionotropic receptors. These are receptors that change function based off of changes to the membrane itself. So let's take a look first at the metabotropic receptor, where upon activation with response to a neurotransmitter, we get a G-link protein activation, which leads to a protein activation, which leads to the opening of the ion channel and movement of ions from the extracellular compartment intracellularly, leading to a change of membrane voltage or a depolarization or hyperpolarization moment, depending upon what type of swing we get. In terms of the ionotropic, we have what are referred to as voltage-gated ionotropic receptors. These are going to respond to changes of membrane potential, where when we flip the membrane potential, they'll go from a closed position to an open position. Most of the voltage-gated channels have a secondary position known as the resting position. It is this resting position that leads to what's referred to as the refractory period that we see with voltage-gated channels. In the ligand-gated channel, we go from a closed position to an open position based off of the binding of a chemical to the transmembrane protein, leading to the conformational change and opening of that protein. And then we get the mechanical gated or the mechanogated channels. This is due to a stress being placed on the membrane itself, which leads to the opening of the channel and allowing for the ions to move, where the channel gate itself will stay open as long as the stress on the membrane remains. So let's take a look at particularly these voltage-gated channel responses as it relates to establishing the membrane potential.
When the cell membrane is at its resting membrane potential, the activation gates of the voltage-gated sodium ion channels are closed and the inactivation gates are open. Voltage-gated potassium ion channels are closed.
So we have this movement of ions that's going to allow for the depolarization moment, but we have to somehow get the membrane back to its resting potential. And the flux of ions through leaking channels is not fast enough in order to allow it to happen to allow for continuous neuronal functions. In this case here, we have to use the sodium-potassium pump. This is going to be utilized actively to repolarize the membrane so that the refractory period can be shortened to allow for continuous action potential trains to be passed along in the action potential pulses. So let's take a look at how the actual sodium potassium pump will function to allow for the memory potential to be reestablished.
In living cells, the concentration of sodium is much greater outside the cell than it is inside the cell, and potassium is much greater inside the cell than outside. The sodium-potassium pump is the main mechanism used by cells in order to maintain the concentration gradient of these two ions. It functions by binding 3 sodium ions, followed by ATP indicated by the star. ATP hydrolysis phosphorylates the pump and induces a conformational change. The phosphorylated form has decreased affinity for sodium, allowing the sodium to diffuse away. The pump binds 2 potassium ions outside of the cell, which causes release of the phosphate and induces a conformational change, which then releases potassium and allows the cycle to
The stimulation of the membrane will lead to the depolarization moment that we just saw. If we get a strong enough depolarization moment, then we get what's referred to as threshold happening, at which point in time the voltage-gated ion channels will open, allowing for the propagation of the potential. This is what's referred to as the action potential. Action potentials will function off of a all or none principle. The all or none principle stipulates that the neuron within the nerve must reach threshold in order to fire. There's no partial response within this. We can get a graded response within the cell body itself, but if we do not reach threshold at the axon hillock, we cannot get an action potential. That's what we mean by all or none. If we get a stimulus that goes beyond what it takes to get threshold, the signal itself doesn't get stronger. However, the amount of action potentials will increase. So we'll go from having a single action potential to multiple action potentials. This threshold potential is approximately a 15 millivolt positive change. And so if we get this plus 15 millivolt change, we go from having no response to having action potential response. Notice that if we go above that threshold stimulus, the action potential does not get larger, but what it does do is it does lead to a larger amount of action potentials. The neuron itself has to go through refractory in order to get to a spot where it can go about doing the action potential cycle again because of the difference between latency and refractory. Muscles will not go through this refractory period. And so what ends up happening, as we get more and more stimulus happening, we go from having single responses to multiple responses to what's referred to as a train of responses. This train of response is how most neurons are functioning at all times. The trains centrally within the cerebral cortex get compressed within the neuronal networks by what's referred to as a logarithmic compression rate, thereby reducing the total output to a manageable signal response. And so the cycle will continue and continue and continue. And we need it to continue because we're constantly having stimulation